noticing the
Hey, what is that,
folds.
that white flash there, right in the...
Wait, let me get my pointing stick out. Oh yeah.
Is that something different? Looks brighter white.
Oh, down there.
Do you guys see that in the back, or is it just the same, just
Yeah,
a
I see
smaller
it.
version of it?
I see it. It's too small to resolve.
Yeah.
Yeah. Might not just have gotten yellowed yet.
Yeah.
Little baby sponge.
Yeah, you can see all this sponge debris here too. So definitely this has been a place for sponges for a long time.
Looks like Argus is gonna be about settled out. I'm gonna change to 120.
2-0, Roger.
We'll be on kind of like the north side, but still going up. And 120, you can see it's still just going up slope.
Did we use the term sponge debris? I don't. Someone's asking, what is sponge debris?
I did, yeah, I was circling that on the Telecaster. So just dead sponge spicules piled up.
Telestrator, not caster. They're actually two different things.
Thank you. I am sleep deprived and not picking my words
That's
thoughtfully.
perfectly understandable.
Are the spicules of the sponges almost always silica down here?
Yes, there are other kinds of sponges. There are different types of materials sponges can make their bodies out of. Most of the ones we find down here are glass sponges.
Can we get a quick zoom on this?
But they can also use, some of them in shallow water can use calcium carbonate, and others just use protein, spongin. Among like the demo sponges, I think have some of the greatest diversity of
Hey, what's going
materials.
on with the USBL hits right now?
Is that a crinoid,
You gonna zoom in there, Jeff?
the yellow one?
Looks like a comatulid, even though it's on a stalk.
Gotcha.
So this is one of the groups that is not,
Nice.
the primnoid underneath.
Rennie, are you on SPL?
However,
Now I
there's a
am.
yellow coral fan just below the bubblegum coral.
Yeah,
right there.
if you can pan down just a little bit, Jake.
Okay.
No, you're moving around.
In a hot second, Jake, you're going to have to pick up, though.
I have to come up.
Okay,
Yeah.
that's fine.
Alright.
In a hot second. It's okay.
There's a gastropod actually on
Oh yeah.
Yeah.
Oh,
one
gosh.
of the pinnules. Lots of organisms graze on crinoids.
Nice.
Great shot.
Yeah,
We can
good
pan
job.
just a little further right over to that bubblegum coral and see the yellow coral that was next to it. Great shot.
Yep.
Yeah.
Down a bit.
Paragorgid here. Most likely a paragorgid, not a macroelium, I would think.
Very few polyps out.
These are the ones that are tough, but
Alright.
yeah, it looks more like a bubblegum coral.
Right. Can you come on to Jeff? I gotta pick up, but catch up.
All right.
We didn't quite see that yellow fan.
Oh, okay. Sorry.
It's okay.
If-
We're going
if
to have
there's,
to start boogieing, guys.
Yeah, I think
Yeah.
we have to get moving.
You have
Jake,
your
are
auto
you
XY one.
Oh, it is?
Yeah.
That's why. That's
I was
why
like,
I
what? I went to go reset the DVL and was like, oh, can't do it.
thank you for that, Rennie.
Yeah, yeah.
For
I
the
guess
front
I was a
row
lot
team,
faster.
a question.
Mhm.
Jake, are you considered a pilot in training, or are you a full-blown pilot at this
I
point?
am.
Full-blown pilot.
All right.
Yeah, yep. I don't know. I guess.
Well, you are now.
Yeah.
You're doing a great job.
So the long stringy things standing straight up or in somewhat bent shapes, I believe those are little bamboo coral.
These ones here actually are, I think, Candidella, so it's a primnoid. We kind of left the bamboo, the unbranched bamboo field. Actually, Scott was just saying that he feels like he's on a polyopogon farm with all these sponges. And it's true, they have some substantial density right in this area. Kind of lost those bamboo whips, unbranched bamboos now.
Another roll that we're going to see in Argus soon.
roll.
There it
There
is.
we go.
Yep.
Jeff, for our audience at home, do you mind putting on the
Yeah, boogie a
one of the
little further
cameras
ahead
from the
there,
back
Jeff.
deck? Yeah, great.
Watch that horizon move. Get a sense of activity.
That last move, Jess, was 120. Looks like
120,
we're
Reg.
starting to go in towards that rather than 135.
So it looks like the surfaces of these rocks just to our left are also probably have those barnacles on them again. You can see those like white specks.
Polyopogon farm, huh?
Yeah, I was. I'll mention that too.
Yeah, so most of these corals, barnacles, and sponges are all filter feeders. And yes, they would be consuming primarily marine snow. There's not a lot of zooplankton at this depth. Just not a lot of live zooplankton. Although I suppose that's a question for the water samples as well. What kind of microbial planktonic activities in the water?
Yeah, there's definitely some living microbes in the water in addition to that might be hitching a ride on the marine snow.
Yeah, so What time is it where we are? We're on Hawaiian Standard Time. It's 11:35. Our watch is about 20 minutes from ending.
See if we can break 1,800 meters before the end of our shift.
Hear that, Jake?
A student from Yakima, Washington is wondering if anyone in the van has ever observed a vampire squid.
Yes.
Tell us more, Rennie.
It's really cool. I was in Revilla Higueto, off of Revilla Higueto Islands in Mexico, and we happened to have a low light camera with us at the time. So we were experimenting a bit with approaching with our lights off, just to see what, how it would react, and it kind of just didn't react for a while. It was really cool. And then some perturbations of the water and with the thruster wash, and really, really pretty display. There's definitely highlight footage of that. I would highly recommend watching it. It's really cool. Dark purple, volcanic rock surrounding, and then the low light camera really made it cinematic.
Yeah, if you search EV Nautilus vampire squid, it comes up on YouTube.
That was Dr. Brennan Phillips at URI. It was his low light camera.
Okay.
Prior to that, he was a Hercules pilot.
I didn't realize that.
He was the ROV manager, wasn't he?
Yeah, he was the kind of ROV operations lead for OET for a bit.
And he actually started as an intern as well, way back.
And now he's a professor. Yep. Look at that.
Oh, the places you'll go.
Yeah. You could tell though he was in the back row, and he was itching. He was jonesing for the front seat. Wanted to be driving around, but he was resisting.
Once a pilot,
Very
always
patient.
a pilot.
Yeah.
I don't know, there's some days I'm tempted.
Yeah. We'd
Go
never
drive
leave the bottom.
ROVs. What?
We'd never leave the bottom. I remember, where were we? What was it? Jarvis or something? We had to peel you off the bottom.
What?
We were, like, Argus was just pulling her cup, and you were still shouting out the names of everything. 'Cause we came upon this. I mean, the scene was incredible with corals.
Maybe.
Was that a little Paragorgia back there? The little
Yeah, I think
one.
it's, the next to the sponge.
Yeah.
Every minute down here is precious.
That was when we were seeing some really spectacular features.
Can we hold up a second? Or
What do you want to?
lower right corner, there's a branched bamboo.
This?
Yeah, that, just below that. Yeah. So it's in the frame, 6 o'clock, bamboo coral.
Okay.
Nice
Right there.
braking action, Jake.
Yeah. And we're going to want to get a close zoom
Okay,
on
sit
that.
down for a second.
Put the e-brake on for that.
No, it's not a sample, but we just want to get
Okay.
good imagery of it. Yeah, that one. Yeah.
Is the branching pattern interesting to you, Steve?
It's the sparse branching pattern that's interesting. Yeah.
Oh, what's
It's got these? I think these are what Scott would call volcano polyps on
Yeah,
this
they're
one.
very like bulbous.
Yeah.
Are the
Are they-
polyps closed right now?
Yes. Yeah, they're retracted and contracted.
Oh, interesting.
Yeah, so it's really tough because the polyps are actually blocking the nodes on this one. But I think it's tough to tell. Could we rotate around to the right a little bit?
Try it.
You're falling backwards. If you
Yeah.
watch your porch cam there.
Sorry about that. Come on, a little bit.
Okay. All right. If we need to move, go ahead.
Yeah, I
Okay.
think we might need to pop in front. starting to see some.
I've been trying to keep an eye out for these branching, kind of sparsely branching bamboos. There's a specific type of sparsely branching bamboo that we're looking for to get imagery of. But it has to be really tight images on the polyps and branch points.
Little tiny primnoid. What appeared to be that.
There's a big fan coming up in the right side. Herc's view. You can also already see it in the Argus view.
Go ahead and push out ahead if you can there, Jake. And then that way when they want to set down.
Yeah, more time. Roger that.
Plus there's a good feature coming up.
Deceivingly big there. Those lasers are telling.
Four more meters to go.
How much time we got?
We got
Pilots will
18
start
minutes.
to come in first, I think.
We'll probably start seeing our counterparts coming in the next 15 minutes for our watch change.
Can you guys go back downstairs for a few minutes? We just have to get to 1800 meters. Thanks.
What's that?
You can tell them to go back downstairs for a few minutes so
Steve's
we can
not
get to
ready to
1800 meters.
See, this is
What were you saying about,
Yeah.
peeling?
I don't have a problem.
I don't have a.
Everything's okay.
Yeah.
Well, we also have an audience that's been with us for quite a while, so
That's right. We
you
have
have fellow compatriots who just love seeing all these animals.
around the world: Australia, Belgium, Brazil, Switzerland, Spain, Hungary, Ireland, Japan, Luxembourg, Norway, Germany, Netherlands, the UK, and a few from Canada and the United States.
One more meter, Jake.
One more.
We're going to do it.
So this is our kind of arbitrary endpoint, but we're going to go far beyond this, most likely. Right?
Yeah, no, 1800 was just like, can we, just a random depth.
Oh, I see. Yeah.
We're trying to entertain ourselves.
Yeah, we've already passed the original endpoint for this dive, and we're just keeping climbing to see what we see.
Yes. There it is.
What?
1800.
Mm.
1799.8.
And boom,
There we go.
1800.
We did it.
I'm gonna
Great
keep
job.
trucking along here. Add another
Yeah,
step.
we're gonna, this, you
Oh,
know,
is that another one of those lithoid crabs there on the, like, 5 o'clock?
Yeah, can we take a look at that? I think that section, that one
Or is
might
that maybe a
actually be
squat
a
lobster?
Which one?
Galathiid. So it's at far right corner, 5 o'clock.
Over here?
White crab.
It's
no,
actually
below.
just out of frame. Oh, there it is, back in frame.
Yeah, I think
Oh, there it is. Okay. I couldn't
think
see
that
it.
that's a Galathiid.
Galathiid.
Oh, yeah. It looks like there's another
Yeah,
one.
I'm
Yeah,
reasonably
that's-
certain we did see some lithodids earlier because, but no, I know we've also seen Galathiids. This one looks like a Galathiid from a distance.
Quick zoom there, Jeff.
Yeah, that's a Galathiid squat lobster.
Dropping these guys here.
Sorry. Come by there. Sorry.
Hate it when you're right, Steve.
Maybe.
He doesn't.
Steve likes the ones
What's
that
that?
are occluded by the sponges.
Trying to figure out what the crab is. What are you making jokes about?
Nothing.
We love it
look
when
at-
you're right.
Look at that sponge that's like in the, it's found a
Yeah.
little spot inside a little lava rock. Crab is.
Anyway, probably maybe Munidopsis.
I haven't seen too many of this really large type of sponge that I see on the left and the right-hand side of the screen, except for here. So is this more kind of endemic to this part of the world? Up north, I don't recall seeing these types of sponges as much.
That'll be a great question for Chris when he comes in the van in a few minutes. Oh wait, no, Chris isn't up next. It's Emil and.
He was saying something about that this could be some sort of, maybe some sort of range extension, but it's not really clear because it's tough to tell them apart from imagery alone. You really have to get a specimen. So they might be. But no, Poliopogon is a genus that you can find in the Atlantic, Pacific. Different species, though, will have more different ranges.
I think you're going to approach a bit of a wall there, Jake. You want to push out ahead a bit?
Double rainbow.
So we're coming up onto some steeper terrain here, which you can see in Argus's camera, as well as in the view of the sonar that's showing up in channel 3, the lower left part of the quad.
A viewer has observed that there doesn't seem to be a lot of visible marine snow in this region, but there must be enough to sustain such a abundance of organisms.
Yeah, when we back away from the seamount, you can actually see particles in the water moving by, and we can tell.
Fish.
There's a fish above us.
Fish shadow.
Looks like it's an alpha-branched. We got a new type of bamboo coral here.
Over to the right.
Yep, over on the right. That's a
Can you pan
what we
right,
call
Jake, just a
Isidella.
little bit?
Isidella, or clade I. I don't know if it's I4. Is it I4? Scott went away, so
Yeah,
ask him
Scott's.
later.
Yeah.
But we'd call that Isidella normally.
Isidella. Putting it in the bank.
Add that to your bingo card.
Yeah.
Rodney, how much do we have left on that ship move there?
20 meters left in the ship move, probably at least 30 for Argus. Let me.
Roger. Okay.
Maybe 40.
That'll set us up nicely for a ship change then.
Yeah, I won't call in anymore.
Oh, that's a different type of sponge there, actually. In the yeah 5 o'clock there.
Right here.
Yeah.
Yeah. Can we have a look at that, Jake?
Sure.
At least some imagery of it. Chris can. That one actually it looks more like a.
Looks almost like a tulip.
Yeah, like a. What's that group?
Oh, really nice.
Now it looks like
I'm
a pig
guessing.
snout.
I don't know if it's.
You can also see some sponge recruits off to the side of different sizes.
Okay, got some imagery of that. Thanks.
Nice.
Yep.
Thank you, Jake.
In your
Jeff?
upward boogie, if you could go more to the port side.
Roger that.
Port forward up.
Port forward up.
I like that dance move name, Upward Boogie.
Upward boogie. What was our team name?
We still haven't settled on a team name.
We don't have one.
Oh, I thought it wasn't at leisure and
Oh, duty and leisure.
Duty and leisure.
That
I'm
was a
going with
contender.
that.
I think that's our slogan, right?
Duty or
Not
leisure.
necessarily
Yeah, that
our
could
name.
be,
With-
that's, you're right. That's
With the
team
lightning
slogan.
bolt between the two.
Right. We got to do it.
I like that,
We
yeah.
happen to have an artist on the watch. So.
Team slogan.
You keep
Still need
saying
a
this
team name.
leisure word, and I still don't know what you're talking about.
It's either that or what was the other? Microbes in the front and
Oh, yeah. And sponges on the side. Kind of
all right, Jake, and we're
a wacky
in
mullet.
preparation for finishing up here. Do you want to boogie out pretty far in front?
Roger that. Move on.
Settle
Like
down
a
there.
we
rotten
can make
mullet or
Steve laugh.
something.
We've got some eager 12 to 4 coworkers here ready to take over. Have to pry some of us away. This has been such an exciting dive.
Mm-hmm.
If you had to sum it up in a couple words, Beth, how would you describe our last 4 hours here on this seamount?
Sponge-tastic.
Oh,
A single word.
I hear Shaggy has a song about that.
All right, we're seeing more of these yellow fans, which
Yeah.
actually might be worth a second look
Okay.
if we
I think
time.
Jake's trying to get ahead.
Yeah,
while
so we're
we're-
trying to get ahead for
Okay.
a shift change too.
Yeah.
We had that,
But
that cliff, but
Alright.
it should be.
The other group will have the glory.
Yep.
It's been lovely exploring with everyone here. Thanks so much for sticking with us. This has been our first daytime watch, and it's been really fun to answer questions. Please keep those questions coming. We've got some experts come in to join, take over for the next 4-hour watch while we go eat our lunch. Try to
Yeah.
rest, try to pry away from the screens.
Yeah, and a reminder to our audience that, you know, this is the first time that the seamount here, the Don Quixote seamount, has been visited with an ROV. So you're joining this exploration live as we uncover the diversity of life here in Papahanaumokuakea Marine National Monument.
Mushroom corals.
Kalamehu, any final words for us on this last view for our watch at least?
Yeah, mahalo nui gang. And so just to our communities in, well, across the world, a couple websites and resources that we can look at. The NOAA website, right? The monument website, papahanaumokuakea.gov. And then the co-trustee website is the Office of Hawaiian Affairs, or OHA, OHA.org/papahanaumokuakea for any sort of like cultural historical context, and then some of the, and just more particular information about like the monument and stuff like that.
Yeah, and a reminder to our audiences based in Hawaii, if you are interested in signing up for a ship-to-shore interaction, there might still be some slots for that. So please visit nautiluslive.org under the education tab and look for that ship-to-shore interaction to request some of those if you'd like to talk with us some more.
Yeah, and if you're interested in joining the expedition, please check out the please also check out the Nautilus page for ways that you may be able to get involved. Um, all there, it's a great community with a lot of diversity that helps these expeditions, that makes these expeditions successful. So please don't be ashamed. Please don't be intimidated. Come and just, I guess, rock and come in, if you will. Come and check it out. See how you can get involved. Well, aloha nui, gang. See you guys later. So much.
Mahalo. All right, we're going to start turning off our microphones as we collaborate with our coworkers here and hand over the reins. I'm excited and delighted to hand this over to my colleague, Kim Weaver, SCF for the, remind me again, the name, the tentacular turtles? Tectonic turtles, the 12 to 4 watch. Mahalo, everyone. Thanks, and happy exploring.
Thanks, everybody. Going off watch.
Challenge accepted. Yeah, settling out. We can touch base with the room when we're ready. Figure out our plan.
Welcome, everyone. We are your 12 to 4 tectonic turtles, just getting settled here for continuing this dive on this amazing site. We are going to make sure everybody's settled and getting together here, and then we will just take a moment to observe a ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we're interacting with on this and other dives. This is something that we are doing to honor the location where we are, which is Papahānaumokuākea Marine Monument, and its importance to the peoples of this region. So check around the team. We're going to go radio silent for a moment, and we'll be back with you shortly to continue this exploration. All right, thank you for that.
I also put flowers in my hair to celebrate all of the knowledge that the site has given us today in this wonderful biodiversity in celebration, and to honor the sacrifice that the Hawaiian people are making in sharing this wonderful place and in celebration.
Definitely a site to be celebrated. This is an amazing diversity and abundance of corals and sponges, and it's interesting. We'll be watching closely on our watch to see how it varies as we change depths. We saw in the previous watch a little decrease in the, at least the corals, and somewhat in the sponges when we were in a saddle between mounds. And
exactly, and so we're wondering, we're going to get into another saddle here, we think, looking at the topography, and we're wondering if we're going to see that same relationship. So this amazing biodiversity and density of animals is one of the things we like to see, because we're down here to test different hypotheses of why certain areas are really chock full of density and high populations and others are not. So we're looking at the current directions, what those current directions might be. And then today we were also talking about what might be upstream of the currents that influence this area. Potentially a nutrient source is upstream from the currents that feed this area, and that might be a reason we get a higher abundance of organisms and a higher level of diversity. So these are all things that as citizen scientists you can think about from your living room as you ooh and ah over these amazing creatures. These sponges have been around for 500 million years. They are absolutely spectacular. So when you look at them and you say, oh wow, they look like a whole alien species, they are actually some of the first multicellular organisms to populate this planet. So we're looking at the interplay between the rocks, the nutrients in the hydrosphere, the hydrosphere is carrying them. So we're looking at the interaction of the geology, the biology, the hydrosphere, and the currents at play. And Emil is a expert on the currents in these waters or in the oceans in general. So it's really interesting to get his take on how they might, the ocean currents and potentially oscillatory currents might be influencing the diversity and abundance that we see out here. I wanted to give a shout out to my students at California State University, Long Beach, and I'm hoping that they're tuning in to see this remarkable place and its remarkable biodiversity and in a deep, deep sea that you know, for many years we didn't even know that these communities existed. So this is a wonderful adventure that we're going on.
We do.
Very slow growing organisms, vulnerable to bottom trawl fishing, perhaps in the future to seabed mining. So it's wonderful that these are under the protection of the National Marine Monument.
Yes, very good. And we don't see any evidence of trawling here. We don't see any scars on the rocks, and so that is a really good thing that we might be able to associate with the density and abundance of so many organisms here. We're really sponge rich in this area. That's another thing that we've been talking about. And we see a lot of bamboo or these string-like corals that you've been noticing. So these are all things that we will take into account because the videos made from these dives are surveyed by biologists, and all of the organisms are counted and identified and plotted through 3D space so that we're trying to determine what the patterns are here to understand these communities better.
And Nav, we got a ship move on to the southeast?
Exactly. Just following what has been happening with all the other watches, kind of creeping up this ridge, making those southwesterly moves, moving at a slow speed.
Yeah, this slow speed is nice. We can dwell on, get some great zooms on these huge sponges.
I also understand that we're looking for a particular small white crab that if we see it, we want to get some closer shots of it. Have you heard of this desire from the science folks? It's not in the chat. They told me about it at shift change.
I did not.
It's the small white one that has the shorter,
I'm going to shoot for the anemone
arms.
here. I got a little bit of cross current.
So it is not a squat lobster?
No, it looks like a squat lobster, but it's white and it has shorter arms and the arms are fatter.
Okay.
And it has a pointy head, like a pointy front part. So maybe Eagle Eye Steve there in the video chair can keep his eagle eyes out for a white little crabby thing with a pointy head.
With a pointy head and fatter arms.
There you go.
A fat-armed white crab.
Challenge accepted.
I believe in you.
So it's, sorry, can you describe it one more time?
Yeah, okay. So picture, if you will, a squat lobster, but then make it white.
Okay,
yeah.
Make the arms about 1/3 of the length and 2 or 3 times fatter, and make it have a pointy head.
Okay.
I'm not a biologist, y'all. I'm doing my best here. This is what they're telling me. They want zooms on it, so we'll see if anybody chimes in on the science chat, but that's what I was...
I'll just look for white crustaceans to start.
Yeah, we'll look for them. That's a good start. They don't want to collect it. They just want to see it. They want to, get a real good zooms on that if we see it.
Copy that.
And Emma, was there interest in that pink squat lobster as well? Again, if we see another one of those to get some more zooms on it?
Not necessarily. It's been collected. It's in the Harvard Museum of Comparative Zoology.
Okay.
But I'm always up for a zoom if we've got time.
Yeah, that was just me that wanted to zoom on that pink. Okay.
Yeah, that was just you. You were the
totally reasonable.
Yeah, I'm all about it. I think that pink squat lobster they saw on that last dive was my spirit animal.
Yeah. I think there's a squat lobster or something on the top of one of those sponges in the foreground. See something sticking up out of one of those sponges on the top of the screen, right?
Oh, in the top of the screen?
Yeah.
Can you mark it?
Yeah.
Oh, no, I don't think oh, there it is. Okay.
Oh.
Let me see if I can get there from
you
here.
See him? He's waving to us. He's
Oh, yeah, I totally see him.
Come celebrate me. Come celebrate me.
I'm coming.
I'm coming.
King of the world.
So was there this much life when we first landed on the seafloor?
Essentially, yeah. I think they just came right down on it, so a fantastic dive site.
So it's noon local time here, Hawaii Standard Time, and this dive started at 4 a.m. Hawaii Standard. They reached the seafloor around 5:15 a.m. and since then we've been seeing communities more diverse and extensive than we've been seeing on any of other dives. So,
and Dr. Chris Kelly says this is probably within the top 5 most diverse and abundant communities that have been documented for deep sea communities in the world.
Wow.
So we've extended the dive from a 4 p.m. recovery to an 8 p.m. recovery.
Video, can you give me a zoom?
And
see what that guy is?
the swell that's moving in is giving us a break. I think the conditions we have now will be the conditions we have when we recover.
That is, I think
nice
it's a squat lobster, isn't it?
Nice little easy chair there. What is it's a small coral?
Okay, go wide.
So keep
I'm
your
pretty
questions
far out.
Keep your questions coming in the chat, or on our website rather. You can enter questions in the box on nautiluslive.org, and we are getting some questions. We're just getting situated here after watch change, but we will be with you for the next 4 hours, and exploring and seeing what we see. So as we're getting going here on our watch, we'll go around as we normally do, and let you know who's here. And since we're getting questions about what we're seeing, I'm interested to know what's the most amazing thing you've seen on this dive. I know some of us have been following along. How much have you been following along on this current dive?
Okay, let's try a zoom again, now that I'm closer.
Yeah, I think that's a squat lobster, right?
Mm-hmm.
It's a beauty little guy hanging out there. He's got a good spot.
If you're a squat lobster, are you eating stuff that comes by in the breeze?
Yes, you are.
You've got your claws
Okay, go on.
extended,
Yeah,
looking for treats.
sometimes you can see them. They got their.
You just snag it out of the
Yep,
sky.
pluck it out like a ninja.
I know I've had the discussion before about why they're called squat lobsters, but I don't remember what the conclusion was, or if there was a conclusion.
I don't remember having that conversation. Maybe Chris will chime in, but
I'm going to guess their posture as their...
yeah, we're going to go with posture.
Okay.
Or maybe they're squatting in these, you know, like maybe it comes from the fact that they're squatters in these amazing structures that they didn't build.
Oh, that's a good hypothesis. I like that one.
Or is it because they're
like
small?
freeloaders? Yeah, just kind of...
oh, it could be, yeah, that they're small.
I like the
squatters' rights.
I like the squatters' rights.
All right. Score one for your SCF, yo.
It's my sponge. I think, though, Chris was mentioning that there's probably, in some of these larger sponges, perhaps hundreds of organisms in them.
Wow. I like that.
A lot of squatters.
Can we move bearing 135 for 100 meters? Thank you.
Yeah, this is the move. Spelunking in the van.
So this is our dive that we are doing here. We've been doing several dives. This one started at 4 a.m. We are on Don Quixote Seamount, which is in Papahanaumokuakea Marine Monument here west of Hawaii. And this dive's been going on for a while. We may dive on this feature again in later dives. But we are super excited at what we're seeing. Don't let these sedate voices online fool you. It is a science party on the ship right now, based on everything that we're seeing here.
And ashore, we're getting some help and observations from scientists ashore. The same telepresence that allows us to communicate with the internet audience also allows us to tap the brain power of scientists ashore, and they're all happy to see this dive site unfolding the way it is.
So what is the stuff on top that's growing? Little green hairs.
Yeah, what is that on top?
Data, your mic could use some adjustment.
It might be just fine pieces of biogenic sediment, you know, the marine snow raining down and then collecting on the sponge.
It looked wispy. How's the mic?
Better, thank you.
So I'm Emil Petrancio. I'm one of the watch leads for this section and physical oceanographer. So that's why you're going to hear descriptions like easy chair for some of these biologicals on top of the sponges. So I'm happy to draw on the brain power of the scientists ashore and elsewhere on the ship. But so Andy Collins, our representative from the Papahanaumokuakea Marine National Monument, and I were interviewed for Hawaii Public Radio, a segment which aired late this morning, just before our watch, and should be available for listening at 2 p.m. Hawaiian Standard Time today at the website for Hawaii Public Radio. Just go to the conversation, and we just give kind of an overview of the monument and the goals of this expedition.
Yeah, and one of the primary goals was to see a diverse, high-density community. So we check that off the list today, which is great. And that's why we have great celebrations, because that is one of our goals, is to see if we can identify where these communities are and how many are out there. So it's a sign of success thus far on our cruise. Those are some magnificent sponges.
It's amazing to think this is the same seamount we were on
Oh,
the last dive, but
yeah.
just a different side of it.
Yeah, we're on the northwest corner right now. We were kind of north central northeast on our last dive,
which is supporting what we've seen before on previous seamounts, that it's the western side has the higher level of biodiversity.
But we're also a bit shallower than we've been on other seamounts, just because Don Quixote reaches shallower depths near the summit.
I think you're muted.
You're not on chat, Gabby.
How big is this sponge? I can't count and fly at the same time.
Yeah, so height, it looks to be, I don't know.
2
Everybody
meters.
think of a number.
I'd say
1.5.
I was going to say 1.2 meters tall.
That's big.
It's big.
And about half a meter wide at its widest.
Yeah, I go for that 1.2, 1.5,
Wow.
yeah.
Yeah, estimation is your friend.
Could we get a zoom on this one while we're here?
Yeah.
Get ready, Steve. It's going to be lots of zooming on this watch. Look
okay,
at those.
I think we're getting a little more stable. Yeah, go for zoom.
This was cool.
Let's see what we've got on here.
Wow.
I think I'm good.
Here.
And
you're getting closer to me, so.
And those spicules are made of silica.
Yeah.
Correct. And they're beautiful.
Very fine structure.
Crashing in here.
Oh, awesome. So we're getting lots of great messages and questions from the chat, but we're getting a special message today from a student who was in one of our live interactions during the school day on the East Coast with us, and now they're at home watching with their younger siblings. So this is how it happens. One person tells another, and we're expanding the reach of this amazing exploration that we're able to do. So thanks so much for tuning in, and thanks for bringing your younger siblings along. We'll look for your questions in the chat.
Some honorary turtles.
That's right, honorary tectonic turtles.
Honorary tectonic turtles, yay.
So I'll continue the introductions. My name is Kim Weaver. I am sitting in the science communication chair, and it's my first time on Nautilus, and excited to be here and answer your questions that are submitted through our website. Let us know what's on your mind, and we will get to your questions as we are able to during the dive.
And my name is Andrea Balbus. I am a geologist, and I study, amongst other things, I date rocks. Yes, that's what I do. And I study intraplate volcanism. So these volcanoes that are now dormant, these are seamounts, but they were once active volcanoes on the seafloor, made likely by mantle plume related processes. So we can track those on the plate, the Pacific plate, to reconstruct how the plate moved through geologic time. So if you are interested in the geology, this is what we call intraplate volcanism. And we take rocks from these expeditions, and we find ages for them, radiometric ages, or at least we try really hard to, so that we can reconstruct the movement, the rate, and the orientation of the plate through geologic time. Look at this spectacular field.
This is such a cool field.
Yeah, field of sponges. Beautiful sponges.
So dramatic.
The glass sponges are being studied by structural engineers to determine how they can be so strong with these the designs that Mother Nature has come up with over hundreds of millions of years of evolution have resulted in a very efficient layout of these silicate bodies that can take the strong current flowing past them.
And we see a volcanic sheet flow right in front of us.
Oh, I was just going back and forth a little bit.
Yeah.
If you're watching on the quad or if you're watching Channel 3, you see...
I was going to zig and zag a bit.
You can see our map, and
if it doesn't end up working, though, I'll stop.
Our nav chair drops points, and it says still abundant sponges, more abundant sponges. There's just a trail of points along the map that's like, yep, still got some sponges.
Well, okay, so the ship is moving, what, 150 or something?
We're moving 135 at 0.2 knots, though the ship has been tracking a little bit faster, just keeping track of it. Yeah.
I'm Sarah Bremer. I'm a geologist as well, and I'm sitting in the data logger chair. And I have not been following this dive. I was sleeping. I had my long sleep before this watch. But it's pretty exciting to wake up to Sponge City.
Can you go to a heading of 135 and I'll try and stay in that box?
So our science lead, Steve Oskovich, had mentioned earlier that as we get into, we're getting a bit shallower.
Oh, it's all good.
Shallower as we move southeast, that we'll be going to the depth range for plexaurid octocorals.
Okay,
Ooh,
I think we're going into the depth range of some of his favorite corals. He likes corals that exist at the shallower end of the deep sea coral communities, and so I think he's really been looking forward to this.
Yep, I'm catching up.
Beautiful sponges.
So what about you, front row? How we doing today? And if you could check in, let us know your role on this watch and any thoughts you have about what the other watches have seen.
We're doing excellent, or I'll speak for myself. I'm doing excellent. And, this is Steve in the video chair. And I haven't had a chance to see too much from this dive so far. So I woke up and saw a lot of sponges in on the screen in the mess, and yeah, I knew it was going to be a good watch.
You finally got some sleep.
Yeah, I would have been sleeping, but I got up for an interaction with Nell Herman's class in Blue Hill, Maine. Nell was a science communication
Coral
fellow
to the left. Coral
in 2014.
There is something pink underneath you, Gabby.
Oh, there he is.
A little hidden coral there.
Little hiding coral.
Little hiding coral.
And one directly
You thought you were going to get away.
So those laser dots are 10 centimeters apart. They are parallel lines stretching out that help us to determine the size